due to the proteoxic stress in immune cells, cardiomyocytes, and neurons. He has authored more than 280 peer-reviewed journal articles. He has 72 filed patent disclosures. 21 of them resulted in companies. Now, may I request Professor Jay Kumar and Professor Sundar Manoharan to chair the session.
This is a very unique session where both the chairs are speakers as well. So we have decided to mutually exclude ourselves from taking time. Dr. Jayakumar, my old friend from the IIT Chennai days, he has agreed to push his talk to the next session. And I will try to limit my talk only to 8 minutes, not the 20 minutes. Make sure that we adjourn for lunch. This is one of the work prompted by my little interaction with Dr. Samuel Raj as well. So I would like to run through some of the findings of what sort of formulations we have made for oral, intravenous, and transdermal formulation. This is specially aided by a HLB nanoparticle matrix for linezolid drug delivery and mainly to combat the antimicrobial resistance. Can somebody help me? Excuse me. No, is this correct? This is on. No, but still it's not. Can you do this? Yeah, I think I can work with this. Okay, let me contextual place where what sort of work we are doing. This is the timeline of all the key antibiotic resistant event that has happened. And we are actually looking at linezolid, which almost had the shelf life of just 1.5 years. And today, the present antibiotic categories depend on imipenem and dime antibiotics in particular. So I will just show 1 or 2 examples of how we can repurpose the linezolid which was phased out, and also try to show how we can repurpose the present antibiotics before it can develop resistance. Next slide, please. Yeah, this is the linezolid, which has a lot of attributes to it. It is bactericidal, it is bacteriostatic, it can decrease the toxin production in gram-positive pathogens, metabolize via hepatic oxidation, and elimination is through different forms, non-renal, renal, and fecal mechanism. And this is a very interesting molecule because you have the chirality here, which is very specific for AMR activity. And then this also has a fluorine moiety attached to this protecting layer, which is also important for the antimicrobial efficiency. And this is having a 5S configuration, which is necessary for antimicrobial resistance. What linezolid does is to inhibit this pathway from synthesizing the 70S initiation complex. Since this is terminating, this happens to be a very productive antimicrobial candidate. Next slide. Okay, now we have tried on 3 different lines. Transdermal, I'm not going to show the transdermal part, but the medical patch as such, which is spun on the medical textiles. And then we also have 1 or 2 examples on how we can take it to oral capsule or intravenous suspension. Next slide. Okay, we have a lot of work going on electrospinning, and that's where we stumbled at this invention. But this is typically how the HLB nanoscaffolds with special architecture is made. This is not crudely synthesized electrospin nanofibers, but this is rationally synthesized. And the proprietary is because it's a very unique formulation where just like your nanoparticle, which has a core shell morphology, we can actually go for a core shell drug delivery, which is encapsulated by both lipophilic as well as hydrophilic moiety that's coming from the PLG as well as PVA, which is very selective. And also the composition of the HLB and PVA matters quite a lot in this sustained drug release. Next slide. Let me quickly show you some of the attributes of this nanoscaffold that we have made. And this is the skin morphology that we have immediately tested for a burn wound model. These are second-degree burn wounds that are inflicted, but you can see several controls are there, and these are the 5, 10, and 15 microgram of linezolid embedded in this nanowires. And you can clearly see that with just 15 microgram of this linezolid, you can almost eliminate the scar and full hair growth and complete healing. Typically, on the 14th day, even the scar withers off on its own, even without peeling. So there are some incentive that we can draw from this. It is effective against resistant strains like MRSA, VRSA. What is important here is linezolid is prescribed only for gram-positive. But what we have found here is that it is in the nanoform, as a nanofiber, linezolid is having more response both for gram-positive and gram-negative. And also it induces hemostasis due to the nanofibrous structure and encapsulates the antibiotics and analgesics through co-spinning. And what you can see here is the drug release level over a prolonged period of time. And that makes this a very interesting patch because you have a sustained release. And this patch has been even tried on vacuum wound healing as a patch. So if you're having diabetic ulcer or if you're having cellulitis, in fact, this particular work was done in the hospital at Karunya University where I was the Vice Chancellor earlier. And we have found that we could even save a 84-year-old lady from amputation, mainly because we could use this as a viable patch for vacuum wound healing. Next slide. This is the cartoon that shows about the same theme of experiments that we conducted. This is the histopathology showing the effect of HLB-based linezolid, which we have done it on burn wound healing. You can see the restoration of the cells, the fibroblasts, and the number of blood cells that are restored before and after the healing. And there are few things that we can draw from this. Increased number of blood cells, pronounced re-epithelialization, and increased level of hydroxyproline level that we can try to achieve out of this. Next slide. And this is the results from the immunohistochemistry. This is for the COX-2 level. There was a question about inflammation, effect of inflammation, and this scaffold comes to be a very viable alternative because it is not only antibacterial, but it's also anti-inflammatory in nature. So you can see a enormous suppression of COX-2 and INOS between these two cartoons and these two cartoons, and this is for over a trial period of 0 to 14 days. Next slide. And we have also made a comparison between the patch that we have made compared to silver-based wound dressings because we want to bring this as a viable alternative for silver-based sealants or silver-based antimicrobial dressings. And one of the reasons why the silver-based one cannot be used for second-degree burn is it directly goes through the liver and seems to hamper the metabolic activity, especially for the pediatric use. And with this in line, we tried to develop this, and we have made one-to-one correlation We have seen there is no side effects on liver in particular, and doesn't disrupt the metabolic activity. It is effective against hospital-acquired MRSA as well, and also it is very good for pro-inflammatory cytokines. Next slide. If you look at the market need, we need to come up with many more formulations. And today, for antimicrobial dressings, there is a great need, and there is also a need for hydrogels, and there is also a need for the traditional adhesive bandages. What we have come up is another formulation which is not only casting this nanoscaffolds on medical patch, but we have also translated this in a hydrogel, and you can see this, the restoration and the cell migration through polymeric hydrogel membranes are very effective. This is actually now going through preclinical trials now. We have tried to incorporate this nanoparticles and nanofibers into this hydrogel, and this is a work that we are initiating with Technion, Israel. Next slide. If you look at the antimicrobial dressings per se, from the 2017 mapping, there are nearly 139 patents, of which only one is available from India. Therefore, there is a great need for antimicrobial dressing, antimicrobial formulations that are needed for combating this drug resistance. Next slide. This most of the work on hydrogel and on the oral formulation is done in our research center. This is called Dr. J.P. Research Park, and I will show you the animal house where we have done this work. Next slide, please. We also have BSL 3 grade animal house facility for infectious disease model. Next slide. This is our operation theater. Next slide. Yeah. The other one is about the oral formulations. And here I want to take the example of imipenem, because that is one for which the rat model has been tried. But the philosophy here is to attack the cell membrane. And if you're making the nanoparticle, the size of the nanoparticle has to be below 100 nanometer. Otherwise, it's not possible. But as of today, the state of the art reports are there with nanoparticles of the order of 140 nanometer. But this is the first report where we are showing the nanometer size is limited to 80 nanometer, which means both for the gram positive and gram negative, it can permeate through the cell membrane. And this is done using a siderophore architecture. And the siderophores are these scarlet ones, which are ably received by the FPTA, and once the FPTA recognizes the siderophore, it takes in there, and then the cell membrane is ruptured. Next slide. Okay, these are the TM evidence, because one need to know whether it's really breaking through the cell wall. And you can see here, these are the liposomes, these are the nanoparticles, and once we bring it into the contact with bacterial cell wall, you can see the decoration of our nanoformulation, and within 2 hours you can see that this intervention is very effective, and by 4 hours the cell wall is completely ruptured. These are the TM evidences that clearly shows that formulations of this sort is very effective for MDRPA. Next slide. So this is the same thing with a lot more finer details. Next slide. Okay, we are actually scaling up one of these formulation, and we are working with one of the industry, that is Murali Krishna Pharma. This is in the production phase, because a similar nanoparticle formulation is already done for paclitaxel for anti-cancer drug, and we are trying to take this in the same line. So I'll stop here and take some questions. If there are any more questions, we can discuss during the lunchtime. Just one question, maybe. It's stable?
Yeah, it's stable. How long?
The nano patch, we have tested for over 6 months.
Okay.
It stays, there's no problem. And especially the testing platform is more of the drug release, and drug release is intact, so therefore it is not hygroscopic, therefore we don't have to worry about it because this is a PLGA, PGA architecture. And then the next question is about the nanoparticle. Nanoparticles come as a powder, and whenever you want to do infusion, you have to just mix it with saline and then inject.
Yeah, that's my question was in solution state, because it tends to aggregate these nanoparticles if you keep, because many times we are facing problems with the gold nanoparticles.
Yes.
Yeah.
But you have seen the TEM morphology.
Yeah.
We have struggled with this over a period of time, but we have a formulation that clearly gives disembodied, unagglomerated particles. So it's not an issue.
So the cytophore architecture doesn't chelate iron?
Doesn't chelate iron, no.
So
actually, the siderophore is selective for carrying iron nanocores.
Yeah.
But what we are doing is we are completely duping the system by putting the drug inside instead of iron.
Okay.
Yes. So that's the that's the rationale.
Okay.
I think we'll stop and take because we have 2 more speakers. We will interact with you. I will be available during the lunch session. Thank you.
Now I invite Professor Harpal Singh, IIT Delhi, to present his talk.